Hello students, so welcome back to my channel. So we are studying immunology. So let's continue. Today's topic is positive versus negative selection. So the positive selection means, can you recognize self? That means if they can recognize self, the white blood cells, they can recognize themselves, and that is done by those white blood cells that are present in the thymic cortex. They keep those T cells that recognize the MHC, and the rest die. So if they can recognize themselves, then they're kept, and that means P is for positive and P is for pass. Then negative selection is actually, do you attack self, and that is done by those cells, that is done on those cells that are present in the thymic medulla. So they remove the T cells that bind strongly to the self antigen. So it's controlled by air protein. If this protein fails, then it results in very autoimmune diseases. And without air, you char means the four diseases that can occur: candida, chronic mucocutaneous candida, then hypoparathyroidism and adrenal insufficiency, and recurrent candidiasis infection. So if we move on, after the positive and the negative selection, the next topic is IPEX. IPEX is actually a disease that is known as immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome. And that is MAD IPEX. MAD, M is for, it affects the male infants, and A is for the autoimmunity. It results in autoimmune derma conditions, for example, dermatitis and nail dystrophy. And D is for diabetes. So MAD IPEX, it is a genetic deficiency of FOXP3 in which there is immune dysregulation. There are multiple polyendocrinopathies, enteropathies, and it's an X-linked syndrome that affects the male infants and result in autoimmune derma conditions, and it can also cause diabetes. Now we talk about more detail about the antigen presenting cells. So there are four type of antigen presenting cells that we have already talked about. The mnemonic, that is a D bar. D is for the dendritic cell, B is for the B cells, L is for the Langerhans cells, and M is for macrophages. And APC, actually the antigen presenting cells, are the DJ, who has to pick the right CD and play it for the T cells on the dance floor. T cells shake the hand using the signal that is known as the TCR. So antigen presenting cells are actually the ones that have the antigens, that have the CD, and it play it for the T cells on the dance floor. T cells shake the hand using the TCR. So there are two type of signals: the exogenous signals, and then there is an endogenous signal. The exogenous signal present on MHC2, whereas the endogenous signal present on MHC1. And the exogenous signal recognized by the CD4, and the endogenous signal recognized by the CD8. Then the signal 2 is actually the security check. There is the B7, which is present on the antigen presenting cell. It binds to the CD28 that are present on the T cells, that is CD80 and 86 on the T cells. And so just remember that this is a security check. They activate the T cells that produces the cytokines. How then B cells become activated? There is a B cell mediated endocytosis, and the antigen, the exogenous antigen, presented on the MHC2 and recognized by the TCR on T cells, and then the CD40 receptor that is present on the B cell bind with the CD40 ligand on TH cells. The cell secretes cytokines that determine this class switching of B cell. B cell activated and produce IgM and undergo class switching and affinity maturation. Then there is peripheral tolerance. Peripheral tolerance means peripheral means the one that is outside the thymus and the bone marrow, and there is inactivation of the self-reactive T and B cells. And when this peripheral tolerance is lost, that result in autoimmunity. So there are three mechanisms that result in peripheral tolerance. That is the energy regulation, T cell suppression, and apoptosis. And energy means a state when they cannot when T and B cell cannot be activated by the exposure to a specific antigen. This happened because of the absent co-stimulatory signals. So peripheral tolerance is actually the tolerance is an activation of the self-reactive antigen by the T and B cell, and it occurs outside the bone marrow and the thymus, and it results in the mechanism involves the energy as well as the regulating T cell suppression, apoptosis, and all this. Then if we talk about the antibody structure, so the antibody is actually a Y-shaped protein with two heavy chains and two light chains, and they are bind together by a disulfide bond. There is a Fab region and then there is an FC region. The Fab region is an antigen binding site actually. Its antigen binding site, it is made up of a light chain and a part of a heavy chain, whereas the FC region is only made from the heavy chains and is actually the functional part that does not bind with the antigen. It has activate the immune system. So Fab region and the FC region are the two regions, and FC region is just made up of the two heavy chains, and it actually responsible for the activation of the immune system. It does not bind the antigen, whereas the Fab region is the antigen binding site that made the light chain and part of the heavy chain. So let's move on. If we move on, then what the FC region does, it helps in the activation of the complement. So it remember by the mnemonic 5 C's, constant and carboxyl terminal is there. It help in complement binding. It has a carbohydrate side chain and also confer the isotype. So it help in the complement activation and it also help in the opsonization. Then if we move on, then we have the COMB, that is actually the mnemonic for the what FC does. So COMB is actually the complement activation, the opsonization, binding to immune cell, and the formation of the MAC. These all are the function of the FC. So there is FC region and then there is a Fab region. That Fab region is the one that bind to the antigen, where the FC region is the one that just bind to the that just help in the activation of the immune system. Remember the 5 C's, it's constant, it's complement activation, carboxyl terminal, and it confer the isotype. Then there is a hinge region that is between the Fab arms that give flexibility and allow the antibody to bind to antigens at the same time. Then there is some variable region and then there is a constant region. The variable region means that changes, it binds to antigens and it deters the specificity, whereas the constant region is the same in all the antibody type. It determines the type of antibodies. For example, the IgG, IgM, that is a constant, and variable means the specific region. So if we talk about the antibody diversity, how many antibodies are made? So before antigens, the diversity is created by the variable region, VDJ, heavy, light, and then there is also enzymes that are involved, the RAG enzymes and the TDT enzyme that add the random nucleotide that increases the diversity. And then there is random pairing, any heavy and any light chain, the pairing of any heavy and any light chain. So these are all the how the diversity is created in the antibodies. Then there are generic generation of the antibody specificity is after when it is being exposed to the antigen. So there is somatic hypermutation and class switching that cause more variations. Specificity, somatic hypermutation means efficiently maturation and changes FC region safe antigen specificity. Then we have the immunoglobulins. So immunoglobulins are of 5 types. They are actually the IgG, IgA, IgM, IgE, and IgED. GAMED is the mnemonic for the types of immunoglobulin. G is the one that greets the baby, that means that crosses the placenta. It is also most abundant and the main antibody in the secondary response, the antibody G. A is the one that is present in the secretions in the milk, in the airway, in the it is present in the form of monomer in the blood but as a dimer, and J chain in the secretion form. Here is a monomer, and M is actually massive pentamer in the blood and first in infection. It's present as massive is a pentamer, and so it's a strong complement activation. E is for the allergy and for the parasite. D is for the development of the B cells. So if we remember G, E, and D, they are actually the monomers. M is massive, that is a pentamer, and A, when it is present in the blood, it is a monomer, but when it is present in the secretion, it is a dimer. So affinity and avidity are two terms. Affinity is actually the strength of one binding, whereas avidity is the strength of many binding. So affinity and avidity. So before activation of the B cells, after activation, after switching, then there is a G, A, and E, that is A, antibody A, G, and E, that can be remembered by mnemonic age. Then we have two antigen types, thymus independent and thymus dependent. Thymus independent are the one antigens that has no proteins, they have no memory, and they have weak response, for example bacterial polysaccharides. Whereas thymus dependent antigens are the one that are actually the protein antigens. They have the strong response, they have a memory, and they have a class switching as well. For example, diphtheria toxide is an example of the thymus dependent antigen. So let's move on now. So let's talk about the complements. So we are going to talk about the complement in the next video. Thank you for watching. Don't forget to subscribe my channel.